The M13A guidance, recently adopted by the International Council for Harmonization, plays an essential role in establishing therapeutic equivalence between generic and reference drug products. Published on October 30th, 2024, this guidance sets a critical foundation for advancements in the pharmaceutical field. Moreover, the importance of harmonization in global standards for generic drug development cannot be overstated. By enhancing access to these vital products across various jurisdictions, standardized guidelines serve to significantly improve market efficiency. The M13 series consists of 3 parts, with M13A specifically focusing on immediate release forms. Subsequent parts address additional strengths and highly variable drugs. This phased approach is strategic, allowing for quicker implementation and adaptability in the industry. As we delve deeper, the M13 harmonization process comprises 5 critical steps: consensus building, endorsement, regulatory consultation, final guideline adoption, and implementation. Each of these steps ensures a thorough and meticulous development and acceptance of the guidelines. The final M13A guidance is organized into 5 distinct sections, which include general principles and specific topics. Its purpose is to clarify the requirements for bioequivalence studies, with a strong emphasis on design and analysis across different study types. In the course of public consultation, feedback illuminated significant interest in the general principles section. This highlighted its vital role in establishing bioequivalence criteria for regulatory purposes, leading to essential clarifications based on those insights. Furthermore, M13A champions a risk-based strategy to assess whether 1 or 2 bioequivalence studies are necessary, particularly for high-risk and non-high-risk products. This approach is fundamental in maintaining regulatory compliance and ensuring safety. Importantly, the FDA anticipates that the M13A guidance will yield substantial cost savings, approximately $50 million annually, in drug development. By reducing unnecessary studies, this initiative could significantly accelerate the approval process for numerous products. Moreover, proactive training and preparation efforts by the FDA regarding the M13A implementation reflect a commitment to ensuring that all stakeholders are well informed and compliant with the new guidelines. In addition, the FDA identified over 1,100 product-specific guidelines, PSGs, requiring revisions, concentrating on high-risk generic drugs. This strategic move aims to streamline regulatory processes while enhancing product safety and efficacy. Significantly, changes in testing requirements now restrict fasting and fed conditions exclusively to high-risk products. This represents the FDA's unwavering commitment to prioritizing safety and efficiency in drug approvals. The development of a risk framework was instituted to categorize products based on their biopharmaceutical properties and formulation complexity. This systematic approach ensures consistency in the assessment of both high and non-high risk categories. For high-risk products, specific study designs, such as fasted and fed studies, are essential to evaluate their performance under varying gastrointestinal conditions, particularly due to their low solubility. In conducting fed studies, the FDA advises utilizing a high-fat, high-calorie meal. This method accurately tests extreme gastrointestinal conditions that enhance solubility for certain products. Conversely, for non-high-risk products, a single fasting or fed study suffices, as their pharmacokinetic performance is less influenced by gastrointestinal variability. This flexibility in study design promotes efficiency and practicality. Fasted studies are especially favored for medications taken with food, as they assess tolerability where food effects may not be significant for single uses. This understanding is crucial for optimizing patient safety. Furthermore, drugs exhibiting pH-dependent solubility necessitate additional studies when taken alongside acid-reducing agents, as variations in gastric pH can substantially impact drug availability. The FDA promotes flexibility in study design for drug approvals, encouraging applicants to scientifically justify their approaches, particularly when proposing waivers for required studies. This cultivates an innovative research environment. Looking ahead, The FDA plans to publish a total of 8,814 revised product-specific guidance, PSGs, that will significantly impact bioequivalent studies by reducing the number of studies required. This pivotal change is poised to affect generic drug development dramatically. The presentation details the planned revisions categorized into critical, major, minor, and editorial classifications, with most M13A related revisions being classified as minor. Understanding these categories is pivotal in clarifying the scope of upcoming changes. Additionally, revisions have been made to language and definitions regarding study populations to align with M13A guidance. This specifically emphasizes distinguishing between healthy subjects and the general population to enhance clarity in study design. The revised guidelines now specifically identify healthy subjects for studies involving drugs intended for single-sex populations. This tailored approach ensures the collection of more relevant safety data. Importantly, a dedicated section addressing reproductive risks for females has been incorporated, advocating for male contraception when drugs pose potential risks of embryo toxicity. This marks a significant advancement in patient safety protocols. Additionally, safety concerns surrounding embryo phototoxicity have been meticulously addressed. Recommendations now urge the selection of healthy subjects who are not of reproductive potential for studies, aiming to minimize risks in clinical trials. Ultimately, the finalization of M13A guidance streamlines the number of required bioequivalent studies, thereby simplifying the approval process for generic drugs. This anticipated change significantly enhances efficiency in drug development. Moreover, the FDA is committed to regularly updating the relevant guidance documents in alignment with M13A. This ongoing process ensures that recommendations accurately reflect the agency's current thinking on drug products, supporting prospective applicants in their development efforts. Prospective applicants are encouraged to propose alternative approaches to the M13A guidance, provided they submit adequate scientific justification. This flexibility fosters innovation while maintaining compliance with regulatory standards. In alignment with M13A, the FDA is revising the ANDA bioequivalence guidance to incorporate the latest updates, thereby enhancing clarity and addressing industry feedback from the previous draft published in 2021. With the finalization of M13A, the FDA aims to support the industry by permitting a single study for many products, significantly reducing the burden of conducting multiple studies for bioequivalence. Moreover, data integrity remains paramount in determining whether a single study can suffice for high-risk product applications. Ensuring that data consistency and reliability are upheld is essential for building regulatory confidence. The Biopharmaceutics Classification System, BCS, profoundly contributes to evaluating drug solubility and permeability. This classification aids in informing risk assessments and establishing study conditions for drug products. Food effects on drug absorption present a critical consideration in classifying products. High-risk drugs frequently exhibit unpredictable food effects, which influence the necessity for additional studies under varying conditions. Furthermore, the FDA evaluates therapeutic equivalence by examining not only the active pharmaceutical ingredient, but also the formulation and real-world use of the products. This comprehensive assessment adds a layer of complexity to product classification. Generic products can indeed be classified as high risk, even if their reference product is low risk, depending on specific manufacturing choices and formulation techniques. Instances of this include the application of novel technologies or formulations that diverge from conventional standards. Collaboration and early dialogue with the FDA are vital for navigating updates in the risk-based approach. Engaging with the agency fosters compliance and paves the way for achieving better outcomes in product approvals. It is essential to provide justification when studies include only one sex, particularly for drugs intended for both. This highlights the necessity of diverse representation within clinical trials. Guidelines advocate for maintaining a similar proportion of males and females in studies designed for drugs intended for both sexes. This promotes comprehensive data collection for effective treatments across diverse populations. For high-risk products, rigorously conducted studies must encompass both fasting and fed conditions, which are crucial for understanding the drug's behavior and ensuring bioequivalence. Additionally, the influence of clinical relevance and pediatric considerations in bioequivalence studies drives the necessity for adaptable regulatory frameworks. These factors significantly affect product dissolution and absorption outcomes. The role of citizen petitions in shaping FDA recommendations underscores the dynamic nature of regulatory guidance. Applicants may find themselves needing to justify deviations from established pharmacokinetic study guidelines. The FDA strives for a harmonized approach to bioequivalent studies aimed at minimizing redundant research and associated costs. This strategic shift enables generic developers to channel resources into pioneering product development. Transitioning towards an efficient regulatory framework can lead to remarkable savings for the generic industry, potentially amounting to hundreds of millions annually. This transformation presents a promising future for innovators. The extensive effort devoted to formulating specific guidelines involved meticulous analysis of vast amounts of data, including literature and FDA records, all aimed at improving regulatory decision-making. Finally, collaboration among various teams within the FDA is crucial for effectively implementing these changes. It ensures that all facets of the drug formulation process receive the attention they deserve. As we conclude, the M13A guidance stands as a beacon of progress in the pharmaceutical industry, guiding us towards greater efficiency, safety, and innovation. Together, let us embrace these advancements, working hand in hand to elevate the standards of drug development for the benefit of public health.